I want to welcome you to my YouTube channel, and this is Ricky Chisambo. In this lesson, we want to revise biology paper 2, which was written in 2016. That was in October. So in this paper, we'll revise section A, which is short answer questions, which carries 44 marks. So we'll start with the first one. Figure 1.1 shows an animal cell as seen under an electron microscope. So this is an animal cell, and then under animal cell we have these parts which are labeled I, F, H, G, and D. We've been given cytoplasm there. So the first one is identify the labeled parts F and G. So we have F, this part, and the G. So what are these two parts? So this is a very simple question. So we are saying that part F is rough endoplasmic reticulum. And then when we look at G, when you look at G closely, you discover that this is the nuclear membrane. Let us move to the next one, which is B. Explain the functions of the parts labeled H and I. So these parts labeled H and I. Now we cannot answer these questions minus identifying them. So let us identify them so that it can be easy for us to explain the functions. So we'll start with H. So H, when you look at H, that is mitochondria. And then I, so this is the outer part of the cell, which is cell membrane, because this is an animal cell, so that is cell membrane. Now let us look at the functions of each. So mitochondrium, so is where, so this is where respiration takes place. And then the second one, which is the cell membrane, so the cell membrane, the outermost part of the animal cell, we are saying that is responsible for controlling things, that enters and leaves the cell. So it was carrying 4 marks. Let us move to C. Suggest 2 cell parts which would be present in figure 1.1 if it was a plant cell. So when you look at it, you'd find that if this was a plant cell, obviously a cell wall should be present. Not only that, even chloroplast, which is responsible for photosynthesis. So those two are enough. So the first one is the cell wall. The second one is chloroplast. Okay, let us move. Okay, so we move to question 2. Figure 2.1 shows the effect of pH on the rate of enzyme catalyzed reactions K and M. So when you look at the rate of reaction this side, and then we have the pH concentration, and then from there we have J and L. So the reaction is K. Here the reaction is M. Now, what term is given to pH at J and L? So pH at J and L. So the answer is simple. That is optimum pH. So that is optimum pH. So the question, explain why the rate of reactions K and M goes down after point J and L respectively. So why is it that the reaction started going down as it reached the point J? Same applies with reaction M. After reaching L, it started going down. So you find that each enzymes, they have what we call optimum point where they should live, and then when they reach that point, they will start to denature. So we are saying that the enzymes start to get denatured because each enzyme has a specific pH. So after reaction K reached at J, it started to decrease, or it was denatured. So C, name the regions of the alimentary canal where reaction K and M takes place. So we have to identify. So K, when you look at the pH, so this is the pH concentration. So when you look here under pH, what comes on your mind? So this is the acidic environment. And then where can we find acid in the alimentary canal? So that is the stomach. So stomach, that is where you find acid. And then M, so M, the pH tells us to say this is the alkaline. So where can we find the alkaline environment in the alimentary canal? So you would find this in the duodenum. Okay, so let us move to D. Suggest one enzyme which can catalyze reaction M. So enzyme which can catalyze reaction M. Let us see reaction M. So this reaction M, which is found in the what? In the alkaline environment. So okay, so one enzyme. So one enzyme is pancreatic amylase. Okay. State one food nutrient which can be catalyzed in a reaction K. So in reaction K, that is the stomach. Now they've said, pay attention to the question. State one food nutrient. So one food nutrient. So what is that one food nutrient? So the answer is proteins. Proteins. Where can we find proteins? In meat we can find proteins, and other foods where we can find proteins. So E states two factors other than pH which affects enzyme activity. So we are looking at what affects enzyme activity. So number one, temperature. So temperature affects the activity of enzymes. So after reaching the optimum point, the enzyme was denatured or it started to decreasing, meaning that after reaching a certain temperature, it will be inactive. The other one, it can be substance concentration. So substance concentration. Or you can say enzyme concentration. So meaning that these are two answers. Let us move to question 3. So we are moving to question 3. Figure 3.1 shows a cross-section through a stem of a plant which had been previously dipped in a red dye for 6 hours. So this is a cross-section of a stem, and then it was dipped in dye which is red for 6 hours. So when you look at the parts here, obviously it is very important for you to know these parts. So we have N, O, P, Q, and then we have R there. So now let us identify. So N, that is the cambium. And then P, this P, look at it closely. So that is the phloem vessel. And then Q, when we look at Q, that is xylem vessel. And then we are remaining with what? We are remaining with Q, we are remaining with R, and the other one that we are remaining with is O. So O is pith, and then R is cortex. Which labeled part in figure 3.1 would be stained red? So which one will be stained red after the stem has been dipped in red dye for 6 hours? So it is Q. So Q will be stained red. Why? So that is Q. We say Q. Why? Because Q, it is the one which is responsible to transport what? Mineral salts. So let us move to the third one. Okay. Give a reason for your answer in A to above. So like I stated earlier, we are saying that the enzyme, the xylem, so the xylem vessel transports transport water and mineral salts. Okay, so we are saying that the red dye will act as mineral salts that will be transported, and Q will be turned red. Okay, so we move to B. Okay, suggest 3 factors that would increase the rate of movement of the dye in the stem when it is dipped in the red dye. So what will increase the rate of movement of dye? So number 1, so we can say high temperature. Not by just saying temperature, no, you have to include high temperature. And then not only that, you can say that is number 1. Number 2, we can say low humidity. And then the third one, you can say wind speed. So wind speed, or if you want, you can write high light intensity. So let us move to question 2 under B. Name the process that will cause the movement of the red dye in the stem. So what is that will cause? So is transpiration pool. Okay, let us move. Question 4. So question 4 was a very simple question. Okay, so when you look at it, so figure 4.1 shows a certain type of flower. So we have, this is a flower, and then we have the parts labeled. So this is figure 4.1. Identify the parts labeled S, T, and U. So these parts we have to identify them. So let us start with, so this is a flower. So let us start with S. So S, what is that part which is labeled S? So that is the anther. Yeah. And then the other one, which is T. So when you look at it closely, you'd find that T is the stigma. And then U, this is the ovary. Okay, so let us move. Identify the type of pollination that occur in the flower in figure 4.1. So this is wind. So this is wind pollination. Why have we said it is wind pollinated flower? Why have we said it is wind pollinated flower? Because number 1, the anther is outside, and then the stigma is outside as well. So it has hair which traps the pollen grains from the anther. So give 2 features from figure 4.1 which supports your answer in B1 above. So you have to be very careful when answering questions like this. You'll find that if you get question B wrong, then definitely question B1 and then question B2, you're going to get it wrong as well. So we are saying that it has number one, feather stigma to trap pollen grains. And then the other one is that anthers are large to increase the chances of pollination. Okay, let us move to C. So C, state one other type of pollination other than the one mentioned in B1 above. So the other type of pollination is insect. So insect pollination, because we have wind, so this is now insect pollination. Give two characteristics of the flower where the type of pollination mentioned in C1 above occurs. So number one is that the characteristics of such flowers, the flowers should be brightly colored petals. So it should have petals which are bright so as to attract insects. So we can say brightly colored petals to attract insects. And then B, so it should have nectar. So that nectar is the one which will attract insects to come. So insects will be coming to collect nectar. As a result, they will help in pollinating the flower. So nectarize to reward insects. Okay, so we move to question 5. So question 5, figure 5.1 below shows pedigree diagrams for two families A and B, outlining inheritance of a sex-linked disease called hemophilia. So we have family A and then we have family B. So these are parents and then offsprings. Now we have H and J. And then key is very important. So this one, which looks like a 0, is normal female. So these are females. Okay, so that it can help us. So this is female, and then this is male. Okay, and then so hemophilic female, hemophilic male. So this is hemophilic. Okay, here in family B, we don't have. Everyone seemed to be normal. So you find that A1 from figure 5.1, which family has a parent who is a carrier for hemophilia? So now when you look at family A, that is where we find a person who is hemophilic. This person who is H. So H was hemophilic. Now, obviously, one of the parents was a carrier for hemophilic, and definitely it can be the mother. So we are saying that family, so family A. So family A, because we've seen the manifestation of hemophilia here. Let us move question A2. So give a reason for your answer above. So we should give a reason. So the family, we can say the family as a child who is hemophilic. Okay, that answer is normal. The family has a child who is hemophilic. Hence, the gene was passed on by a carrier. Okay, so B, let us move to B. So using the symbols, which is XH, and then this is normal, and then we have XH, which is heterozygous, and then state the genotype for offspring. So we have H and J if J is a carrier for hemophilia. So if J is a carrier for hemophilia, so H, who is this H? So H, when you look at the key, so H is male. So we would say X H Y. So that is, and then let us look at J. So J, so J here, so J is a normal female. So normal female, we can say X H X H, because J is a carrier for hemophilia. So this is a carrier. So this H, which is capital letter, it was dominant. So this is what we say. So H is hemophilic normal, and then this is now H small, it is hemophilic. We move. So using a genetic diagram, show whether the offspring would be hemophilic or normal when H and J are crossed. Okay, so carrying 5 marks. So we are saying that the first thing that you have to write is we write father, and then we write mother, so that we are able to know. So we'll say the first one is parental. So parental phenotype. So parental phenotype, the father is hemophilic H. So the father is hemophilic. And then the mother is normal. So the mother is normal. And then from there, don't forget to put an X which demarcates the two. And then we have parental genotype. So parental genotype. So we have parental male for father, which is X, who is hemophilic, and then the mother is normal, but she's a carrier. Okay, the other step, it is the gametes. So you separate the two. So we separate, so we have X, H, then we have Y there. So again we separate even this one, so we have X, H, then here we have X, H. So the next step is, so we say this is the second generation. So this is the second never forget, because this is where they've taught us to say after they had children, family A and family B, and then the children had to be crossed. So it means that that is the second generation. So we write offspring genotype. Okay, so we'll say this X will go with that, so it will be X, H, and then X, H, small letter, then the same one will go with this one, so it is H. Okay, so this is homozygous, which is recessive. Okay, so we're going to have H. Then what else do we have there? Okay, so we have a male who is hemophilic. So let us write F2 offspring phenotype. Okay, so we have, this is normal female, and then this is hemophilic, so this is hemophilic female. Okay, and then this is normal male, and then the last one is hemophilic. So this is hemophilic male. Okay, so it means that you've gotten a 5 marks. So these were the answers which were required or needed from you. Thank you for watching the video. You can subscribe for more of such videos.